Hello, everyone. Welcome to our fourth AP Bio Chat. So we are like a month and a week-ish out from your exam. And today, rather than fielding a question from you guys, I decided to focus on a topic that I think gives students trouble pretty annually. And this is the chi-square analysis or the chi-square test. Just a reminder before we start diving into things, you can join us every Tuesday and Thursday here on YouTube Live at 2 p.m. Eastern Standard Time, and we'll be reviewing some AP Bio content based either on your questions or based on topics that are perennial challenges for students. If you have any questions, my name is Lina. You can feel free to type questions into the chat, and I'll be looking over there. If you have questions, you can also feel free to email me. All right, so let's get started. So I always start here, just a reminder that units 1 through 6 are going to be on your test. Units 7 and 8 are not going to be tested this year. The exam dates, which were determined as of April 3rd, are right here. Unlike in the past where everyone would sort of take their exam at 8 a.m. their time, because they're administering the exams online this year for security purposes, everyone is taking them at 2 p.m. Eastern Standard Time on May 18th. Everyone will be taking them at 4 p.m. Eastern Standard Time on June 3rd. As we discussed last week, there's 2 free response questions. One is going to be experimental based, and it's going to be the bulk of your score. You should spend about 25 minutes on this part. And then the second one will be shorter. It'll be about 35% of your score, and it's a 15-minute long response to some sort of conceptual question. And just FYI, if you do have any questions pertaining to AP Biology, feel free to DM them to our Instagram account, which is right here. So it's DNA_learning_center. Okay. And then I think last week there was a question that I wasn't sure the answer to. So you have 45 minutes, it's online, and the deal is that you can use your notes, but I think you don't want to have to use your notes since this is such a quick exam, like 40. So I think open note would be like a big deal if they still kept some sort of multiple choice component to this test, but because it's all free response, I think that having notes, you should keep them to the side so that you have like a security blanket, but I think if you spend a lot of time looking through notes, you're just going to run out of time. Okay, so the chi-square test is more simply referred to as the goodness of fit test. It's a test statistic similar to, a mean, a median, or a mode. So it's helping us describe a statistical set of data. And the description that it gives us is how closely our observed data fits what we would expect that data to be. So whenever I teach this, I sort of just use like a little cartoon of your expectations versus reality, right? So chi-square will help us determine if our reality, in this case, deviates far away from our expectation, or if our reality could mirror our expectations or come pretty close, right? So it's determining whether or not differences in the data are statistically significant or not. Okay. And so just like a quick hypothetical, if you've ever tossed a coin or played a coin toss, you know that a flip of a coin is a fair test in theory because you've got a 50-50 chance of flipping a heads or a tails for a fair coin, right? So if I did an experiment and I tossed a coin 1,000 times, if that coin were fair, I would expect about 500 heads and 500 tails to be flipped, right? But the reality of our universe is that there is a random chance of error or deviation away from that perfect 500 heads and perfect 500 tails. So let's say you did this experiment and you counted 550 heads and 450 tails. You might want to ask yourself, is this deviation away from our expectation, is it due to random chance? Is it because of the random error that exists in our entropy filled universe? Or is there something more statistically significant going on, such as maybe you have a weighted coin, right? Okay, cool. So how would we go about answering this question. Is this difference due to random chance or is it due to something more significant? So the steps for using a chi-square test, the first thing that you always want to do is define your hypotheses. In biology, hopefully you've learned about the null and the alternative hypothesis. The null hypothesis, if something is null and void, it means nothing, right? And so the null hypothesis basically assumes that nothing is happening. In other words, there is no statistical difference between your expectations and your observations. The alternative hypothesis is exactly what it sounds like. It assumes that something else is going on, right? And so for this particular experiment, our null hypothesis would say that there's no statistically significant difference between your observed coin flips and what you were expecting. The alternative hypothesis would be that there is a statistically significant difference between your observations and what you were expecting. Okay, so now that we have our hypotheses written out, you would calculate what your expected values were. And we did this already, right? If you're flipping your coin 1,000 times and the expectation is that half the time you get a head and then half the time you get a tail, your expected values would be 500 heads and 500 tails. And importantly, at this point, I'd like to point out that for your expected values, you can either use a frequency, in other words, like a proportion, or you can use counts. It's just whichever one you pick, you have to stick with. So if you wanted to do frequency for this one, you could just do 0.5 heads and 0.5 tails, right? I'm going to do counts for this, so I'm going to go ahead and use 500 heads and 500 tails. And then whichever one you pick, you just have to stay consistent. All right, the next step is to go ahead and calculate the chi-square statistic. So the chi-square statistic is actually a sum. So this is our formula over here, and the AP will provide you with this information. They'll tell you chi-squared is equal to the sum of your observed minus your expected quantity squared over your expected. And they'll tell you what E is and they'll tell you what O is. So this is all going to be given to you. What you will have to construct is a little contingency table for yourself. So I always split my table up into 4 columns, one for your categories. In this case, our categories are heads and tails because we can either flip a head or we can flip a tail. If we were doing the same sort of test for a dice, right, rolling a dice, how many categories would we have? Well, there are 6 sides on a die, or on a dice. So we would have 1, 2, 3, 4, 5, and 6 for our categories. So figuring out the categories is going to require understanding the problem or the question that you're asking. So in this case, we have 2 categories. The second column should be your observed values. The third column should be your expected values. And then the fourth should be your little plug-in of the chi-square statistic. All right, so we said that we observed 550 head tosses and 450 tail tosses. So I've filled that into my chart. We also calculated our expected values to be 500 and 500, assuming a fair coin. And then the last sort of step, right? So this is our O and this is our E. Our last little step is to use the values for each of the categories and plug them into this equation, right? So we're going to apply this equation to heads, and we're going to apply this equation to tails. And when we do that, we end up with the following. So our observed value for heads was 550, so we plug it in here. Our expected values were 500, and then our expected values were 500. We do the same thing for tails. And then we end up with 5 for heads and 5 for tails. Now I want to point out something about the equation over here. Hopefully at this point in your math course, you have seen this capital Greek letter sigma before. The Greek letter sigma, if you have not seen it before, is an operator. An operator just tells you what math function to do. And this particular operator means to sum. So we are going to take the sum. And the reason I like to organize my table like this is because it makes it really easy to add. So all we have to do at this point is take this value and this value and add them together. And plug that in here to figure out that our chi-square statistic is 10. All right, so the thing about chi-square statistics is that the chi-square value means nothing on its own. You have to use it to find the probability that, assuming the hypothesis is true, the observed data set could have resulted from random fluctuations. And so we use what is known as a p-value table. In order to use it, you first have to determine something called degrees of freedom. So degrees of freedom is a statistical measure that involves something called parameters, but for the purpose of AP Bio, you're never going to have parameters. And so the degrees of freedom equation boils down to the number of categories you have minus 1. So in this particular case, as we discussed already, we have 2 categories, right? We could either have a heads or a tails. And so our degrees of freedom will be equal to 2 minus 1. And so our degrees of freedom will be equal to 1 in this case. Our next step is to use our degrees of freedom and our predetermined p-value. Biologists like to use a p-value of 0.05. I'll explain why in a second, to find the critical value. So this is the chi-square table taken from your AP formula sheet. So this is exactly what it looks like. The table you might have used in your class probably involved more values. So not just p-values of 0.05 and 0.01. I decided to show you this one since this is the one you're going to see on your exam. So the way that you use this table is as follows. We calculated our degrees of freedom to be 1. So we're going to go to the degrees of freedom row and look right here. The p-value that we're always going to use for AP Bio is a p-value of 0.05. And so that corresponds to this row here. And so our critical value is 3.84. So a standard cutoff point used by biologists is a probability of 0.05 or 5%. The basic idea is this: if your values run on a curve that looks something like this, the p of 0.05 represents 5% of possibilities that are represented in this sort of like extreme region of the curve. And then this is where our expected value is. And so if your value is at this 0.05 value or below, what we're sort of saying here is that you're not very close. You're too far away, or you're too extremely deviant from your expected value. So this is too, I'm going to go ahead and say that values down here are too extreme. And because they're too extreme, if you get a value down here, a critical value down here, what you're going to do is you're going to reject the null. Okay. So Swaraj just said, please explain the P again. So the P, again, it's the probability of getting results as extreme or deviant as the observed results. So again, oops, if our P is down here, anything in this small snippet of curve is too extreme or too far away from our expected value over here, right? And so we would not be able to accept the null hypothesis in that case. Okay. Our last step involves sort of what I just said. We're going to compare our chi-squared statistic to our critical value. And there are a couple of parameters. So this you need to memorize. So this should be memorized. If your chi-squared statistic is greater than your critical value, we reject the null. And basically that means our data does not fit with our expectations, right? If our chi-squared statistic is less than our critical value, we fail to reject the null. In other words, our data fits well with what we expected. So if we go back to this table here, you'll notice that the critical values increase as you get a smaller p-value. And that sort of makes sense, right? So the critical values increase this way. So the critical values are increasing as you move this way on the graph. And so if the critical value is larger than your chi-square statistic, it's in that extreme land that we would reject, right? Okay, so again, if the chi-square statistic is greater than your critical value, you can reject the null. If the chi-square statistic is less than the critical value, we can fail to reject the null. Okay. In this particular case, our critical value is 3.84 and our chi-square statistic was 10. And so our critical value is less than our chi-square statistic, which fits our first scenario, which means that we can reject the null. In other words, the coin that we were tossing, that difference, so, flipping the 550 and the 450 is a little bit too extremely different from our expectations to accept that this was a fair coin. All right, and so that's sort of an example that isn't really biologically related at all. So I wanted to talk about an application that you might see on the exam. And this would probably be for question number 1, if you were to see chi-square. Because again, chi-square is a test of like, did your data match your expectations? And where do we collect data? We collect it in experiments. And question 1 is an experiment-based question. So in this particular scenario, our question is, does the data match what we know about Mendelian inheritance? So the scenario is this: you're taking 2 plants that are heterozygous for flower color, where purple is dominant to white. You collect seeds from the offspring and you grow them, and you find that 70 plants have purple flowers while 30 plants have white flowers. And so the question is, does your observation of 70-30 match the expected Mendelian inheritance values? So we're going to follow the exact same process. So we always start by writing out your null and your alternative hypotheses. This is really, really important. And so your null hypothesis would be that there's no statistically significant difference between your observations and your expectations. In other words, yes, there's evidence that supports Mendelian inheritance. And then the alternative hypothesis says that, maybe something funky is going on with the data, and it's not just random chance funky. It's like, maybe these genes are not following the laws of Mendelian inheritance, right? Okay, the next step is to calculate the expected values. For Mendelian inheritance, how do we figure out expected values? How do we predict what we would expect in the F1 generation? We use a tool called a Punnett square, right? So if we are taking 2 heterozygous parent plants, we can set up our Punnett square like this. I'm not going to walk you through going through the Punnett square, but our expectation would be that there were 75% purple plants and 25% white plants. Or, if we were growing 100 flowers, which I think is what the prompt said, we would expect to see 75 purple flowers and 25 white flowers, right? And again, this is where, you pick what type of math you want to do. Do you want to work in percent land or proportion land, or do you want to work in count land? Whichever one you pick, you have to stay consistent when you're doing your observed values. And because the problem gave us observed values as counts already, I think it's easier to just go with this. So I'm going to go ahead and do this with counts. So our next step is to calculate the chi-square statistic, simply using our little chart. They told us that we observed 70 purple and 30 white. We were expecting 75 purple and 25 white. We plug that into our little equation over here. We sum down and we find that the chi-square statistic is 1.33. Okay. Any questions so far on this application? All right, great. The next sort of step is to determine the degrees of freedom. See if you can do that before we go over it together. So again, we had 2 categories. Remember, we said degrees of freedom is the number of categories, or the number of possible outcomes minus 1. And so since there were two possible outcomes here, our degrees of freedom will be 1. So we have 1 degree of freedom to work with. And then our next step is to use our little table and our degrees of freedom and our p-value. Again, we're using 0.05. We're going to find the critical value. Our critical value, once again, is 3.84. And then our last step is to compare our critical value to our chi-square statistic. Remember, our chi-square statistic is not very useful if we don't compare it to anything. And so in this particular case, our chi-square statistic is 1.33. Our critical value is 3.84. Therefore, our critical value is greater than our chi-square statistic, and that fits this second scenario. So if our chi-square statistic is less than the critical value, we fail to reject the null. In other words, this data seems to fit well with what our expectation was, and any differences between our expectations and our observations is probably due to random chance. All right, so we are almost at the half-hour mark, so I just wanted to leave you guys with a more challenging question. So what I would do if I were you and I were studying for this AP exam is I would screenshot this, and what I am planning on doing is next session, we can go through the answers to this one together. This is a dihybrid problem. And so that's going to change our chi-square math just a little bit, but it's more or less the same thing. So don't get thrown off if you see something like this. The idea is this, you probably remember learning that a lot of inheritance doesn't actually follow Mendelian inheritance laws, right? And so they might ask you a question that says, you know, are these two genes linked or do they assort independently? So remember, independent assortment is one of the sort of conclusions of Mendel's experiment. So we want to find out whether or not this particular plant is following Mendel's law of independent assortment. So the problem says, in a type of plant, purple flower is dominant to white flower. Okay, so one of our traits is flower color, and small leaves are dominant to large leaves. So our second trait is leaf size. We're taking homozygous dominant plants for both traits and mating them with homozygous recessive plants for both traits. So you should know that the F1s are going to be dihybrids. And then we're test crossing those. And so we'll have a cross that looks like this. If you remember this unit really well, you probably won't have to do a Punnett square to figure out the expected values. So knowing Mendel's experiments really well will come in handy, just saving you time-wise here. But if you did need to sketch out this Punnett square, it's not that big of a deal. So the first thing you would do is you'd figure out what the expected offspring would look like phenotypically. And then these are our observations. So they made 1,000 observations. These were the observed phenotypes. And your job is to determine whether or not the two genes are following independent assortment, or if it's possible that maybe this data supports the idea that maybe these two genes are linked. And just a reminder, if genes are linked, that means that they are located close together on the chromosome, and so they don't separate during meiosis. Okay, cool. One other thing that I wanted to point out, just like as a hint before you take this one on, is that the categories are different this time, right? It's not a coin toss. It's not a heads or tails possible outcomes scenario. We've got more categories, right? And so the chi-square math will be a little bit different. Are there any questions before we close out today? And then again, if you solve this problem and you want the answer key, I can send it to you. You can just feel free to email me. If not, thank you for joining us. Feel free to reach out on Instagram or via email with more questions, and good luck with your studies. Bye.